Hello, everyone, and welcome to today's episode of ESDR Kitchen. I can still see the participants are coming in already, and it's great to have these numbers. You will know that the kitchen is a fantastic output of the ESDR, where we try to bring to you different types of content at these lunchtime sessions. And today we have the sweet and sour element of the kitchen. I will chair this with my co-host, Dr. Remco van Doorn, who is a fantastic dermatologist and clinician scientist in melanoma biology and genetics. And we are privileged to have two world-leading speakers debating the topic of aging. The topic today is skin aging reversible, or is it a lost cause when loss of stemness and increased senescence emerge? And who better to help us around these difficult issues than the esteemed speakers that we're about to introduce. And so now I'll invite my co-host, Remco, to introduce our first speaker, and I will introduce the second.
Thank you, Neil. It is a pleasure for me to introduce Peter de Keizer, or Peter de Keizer in English. He is an associate professor at the University Medical Center in Utrecht in the Netherlands, and also a founder of the Clera biotech company. He leads a research group that studies senescence in cancer and aging. He studied in Utrecht at Harvard Medical School and performed a postdoc in San Francisco, also in the Buck Institute for Research on Aging. In addition to his studies on melanoma, he studied senescence in aging, in particular the role of FOXO4 transcription factors and interference with the function of this particular transcription factor as a potential strategy to eliminate senescent cells, which is also the topic of at least part of this webinar. Peter, we are very interested in what you will share with us. I give you the opportunity to lay out your position in this matter.
Many thanks, Remco, for the kind introduction, and all of you, of course, for attending. Let's see if you can see my full presentation this way. So, can you see my presentation, Remco? Yes, okay, great. So yes. For a very long time, this has been the fundamental research question that I've been trying to address. And of course, it's a huge one, but why do we age? And we see this on the outside. I don't need to tell you that by UV exposure, our skin ages more rapidly on one side, sometimes than the other. This is a fundamental question that I've been trying to address. And I'm of the domain where we think that at least some of the aspects of aging can be explained. I think it's actually the dominant explanation, in my opinion, by DNA damage. So by being alive, there are many, many types of damage that we occur from my diet and from UV exposure, but also metabolism, single-stranded breaks, double-stranded breaks. It goes too far to explain how many there are. But what we've noticed in the late 90s in humans, but then also in mice, is if you mutate proteins that do the repair of this kind of these types of DNA damage. And again, this is as broad as it gets. So you get fast aging humans and fast aging mice. So at least DNA damage is a cause for aging. That doesn't mean all aging is caused by DNA damage. But for instance, you can see in the bottom mouse, that's the one that we use a lot in our lab. It's a trichothiodystrophy mouse model, and that actually has brittle hair and a lot of skin aging. So for today's topic, I think this is a nice example how accelerated DNA damage can lead to accelerated aging. Now, the problem is that if we look at cells of these mice, they actually function relatively well. They're not really in a bad state individually. So there must be something happening on an organismal level, more so than on the cellular level. Now, that brings us to the main, one of the first points I want to make for today. It's based on two papers, actually from the same group. I don't have shares in this group, but I just like both of their papers a lot. Which is that DNA damage throughout aging can cause two things. First of all, it can cause a loss of stem cells. As you can see here, for instance, in the hair cycle, every time we lose hairs and we regain them when we are young, there's homeostasis. But at some point, throughout time, we accumulate damage around the hair follicles and also in the hair follicles, and this causes the loss of stem cells. And then over time, you just have less stem cells, and therefore you have aging of the hair and potentially of the skin. That's one. The other thing is that there's competition of stem cells throughout aging. So again, it's from the same group, but a different story. And what you can see here in this overview is, for instance, if you look in the young skin, you have these red, yellow, and blue stem cells, and they compete with each other. And at some point, one of them becomes dominant. And throughout time, at some point, that means you get loser stem cells. They are faded out, they peter out, and only one cell that remains. At some point, that becomes exhausted. So again, in both cases, what this points out to me is that the result of this is that DNA damage and aging cause a loss of stem cells. And this could then argue, but it's lost, because it's very difficult to regain stem cells. So that is one point that we would like to discuss today. Is that really the case or not? However, what we also realized is that there are things happening in the vicinity of stem cells. And of course, I will go to senescence in a minute. So could it be that there are cues around these stem cells that can still be targeted and that you can then replenish or improve the fitness of your stem cells? And then just a little introduction on senescence. I think a lot of you are familiar with it, but let's say cellular senescence, which is different from organismal senescence, is a state where cells start to communicate in a bad manner with each other. So like I'm doing to you now, cells communicate to one another. If there is irreparable damage, so the damage throughout life becomes extreme and it cannot be resolved, the cell can become senescent. These senescent cells secrete a lot of factors, and this is very poorly defined as the so-called SASP, senescence-associated secretory phenotype. There are many, many factors involved, over 100 proteins and microRNAs, circular RNAs, long non-coding RNAs, lipids, metabolites. That's a whole field on its own, figuring out how this works. But as a consequence, the neighboring cells, either close by, also over long distance, they become confused. They lose their function. So that's a cartoon. And what we recently studied, and it's now under review, and that's basically the second topic that I want to address today, is that indeed senescent cells can do something very strange to neighboring cells. They can make them more progenitor-like, and they do this in a more or less permanent manner. So what you can see here, and I apologize because this is not a skin system, it's a liver system. It's a liver 3D multicellular structure that we used, where we added either healthy cells, just 5%, or 5% senescent cells. And you see that by adding 5% senescent cells, other cells become progenitors. So actually you get an enormous increase in the number of dedifferentiated cells. So senescent cells, they tell neighboring cells to lose function and shift back in their embryonic evolution, which I think is fascinating because it means you have a permanent arrest of stemness. This could mean that if we have senescent cells in our skin or around the hair follicles, because we have DNA damage with aging, that could be something we can target. Now, this is something we already addressed several years ago when we made a compound that we could use to kill senescent cells. And it's a particular type of senescent cells. I will not go into that now, but in this mouse model that I introduced a few slides back, the fast-aging tycho-tyodystrophy model, you can see that on the left, a 26-week-old mouse, a relatively young, happy-year-old, already has brittle hair, and poor skin morphology. But after 3 weeks of treatment with this compound, there was a lot of hair regrowth, and we see this consistently. And it was also, we think there was also an improvement of the skin, but we didn't study that in great detail. Now, to come back to this, the system of the stem cells, we also applied this trick again in vivo in our mice. So we used a new compound, so that's an improved version of the one I just showed you. It's called CL04183. It's just a number, but let's say it's a compound with which we can specifically kill scarred senescent cells. Now, if you give mice a drug called CCL4, what you get in the liver is two things, and I think something similar happens in the skin. That's why I show this. So if you have extreme damage, you get fibrosis. So you get a lot of smooth muscle actin, and that's actually the consequence of senescence. There have been tons of papers showing this. What we observed is that directly around these fibrotic tissues, you get CD44 upregulation. CD44 is a biomarker for progenitors. Now, if we then remove the senescent cells, what we see is we do not lose the fibrosis, so that stays. However, we do get a strong reduction in the number of progenitor cells. So CD44 goes down. So they can say, okay, so what? Well, that is very much tied to function. So as we can, we also looked at functional biomarkers for liver, and again, I think it will probably be the same in the skin. That I am arguing for this. If you now look at albumin, which is a marker of function of hepatocytes in the liver, you can actually see that it is completely restored in these mice. So in the tortured, damaged mice, only 35% of the hepatocytes still had their function, and we could increase this to 70% by removing the senescent cells. So again, to sum up, DNA damage can cause aging, DNA damage can cause senescence, but senescent cells do something strange. They make neighboring cells lose their function by pushing them into a stem cell state, and killing those senescent cells can actually restore function by undoing this. So could this mean in the skin that indeed by removing senescent cells, that now you can actually tell stem cells or neighboring differentiated cells to actually perform better? Now, to do this is the last two data slides that I have. We actually outsourced an experiment to a lab in Italy from Professor Marco Malavolta, who had very old mice, 27 months of age at the time of start of the experiment, so geriatric mice. If we now apply this compound, the anti-senescence compound, once a month, and we gave them 3 shots, so 3 shots and then a month off, 3 shots, a month off, you actually see that there's lifespan extension. So again, these mice were already very old at the time of the initiation, and still we could get a very nice lifespan extension in these mice. However, and that's I think interesting for today, also in these mice we saw that there was a very strong improvement on fur health throughout time. So you can see here some examples that of which they took images of the mice. So the control mice, they all lose their hair and they start to crouch, and the skin becomes brittle, and they made a fur frailty index for this, which I won't go into the details of, but you can see that there's 2 mocks here, the blue and the green line, and you can see that skin fur frailty actually goes up throughout age. But in the treated animals, already after 1 month, it looks better, and it actually stays much better for longer. So especially in the beginning, you have a very big gain in maintenance of skin and fur frailty. So indeed, it seems that by killing senescent cells, not all is lost. So we can still maintain skin and fur intensity to an extent. So I think my conclusions are the following. At least to an extent, skin and hair follicle aging is associated with a loss of stem cells. And that may be hard to change. That loss of stem cells will lead to a permanent change in the epidermis and the dermis, and that may be hard to reverse. But not all is lost, in my opinion, because cellular senescence can occur around other cell types, and that forces them in a state of progenitorness, which prevents their function. But this is also not permanent, because you can kill these senescent cells by various compounds, including the 4-1-8-3 compound that I showed you, and that can extend fur density and potentially even reverse the phenotype. We're looking into that. So the 2 topics I would like to discuss is the loss of stem cells. Is that the dominating thing we think that occurs in human aging? So is loss of stem cells permanent, and therefore it's still very difficult to reverse skin or hair aging? Or is it the second point, that senescent cells around stem cells or around hair follicles, that they are the bigger problem, and that can be reversed? So this is what I would like to trigger in the discussion, and just a small thank you to my lab for this. Thanks.
Thank you, Peter. That was fantastic. And I'm about to introduce the counter to that very provocative and exciting talk. But before I do that, I just wanted to maybe highlight one of the ESCR board members on today's topic on LinkedIn, Professor Sandrine Dubrac, who said, today we will have the Pope of Senolytics and the Popes of Aging discussing things. And truly, it seems to be exactly that. So without further ado, I want to invite Professor Pidder Jansen-Duerr, who is the Professor of Molecular and Cell Biology and Head of Institute of Biomedical Aging Research at the University of Innsbruck. His lab works on the role of mitochondrial function and dysfunction in aging processes and cellular senescence. And it is a real privilege to have your counter, Professor Jansen-Duerr, to the provocative and exciting comments from Professor Pidder to Keizer. Please.
Yeah, thank you very much for the invitation and for the nice introductory words. I'm not sure that we are really running into a clash because, I mean, I know Peter's work since a lot of years, and my feeling is instead of going apart, we maybe meet somehow in a quite interesting niche. Having said this, I would like to share my my slides. I can do this, yes. So my topic is on the reversal of skin aging by elimination of senescent cells. In the Hallmarks of Aging that has been published a couple of years ago, one of the hallmarks of aging is cellular senescence. So organismic aging is due in part to cellular senescence. I show you here that cellular senescence can be induced in human cells by repeated passaging, which leads to progressive telomere shortening and subsequent cell cycle arrest. Alternatively, premature senescence can also be induced by stressing the cells, for example, by UV irradiation, but many other procedures. And what is clear now that senescent cells accumulate with age in many tissues, and there is convincing evidence from mouse models that genetic elimination of senescent cells delays aging. I can't go into much detail here. As already alluded to by Peter, there is a new trend in the field to try to eliminate senescent cells by applying small molecules, not genetically, which would not be possible in humans. And these compounds are called senolytica, which may allow clinical translation in the end. And in the next slide, you will have a déjà vu. I have chosen this very impressive senolytic molecule developed by Peter and his lab. And here you see the functional and phenotypical rejuvenation, which we just have seen by a senolytic peptide. But now there are many groups working on senolytics, and senolytics are being tested in mouse models for a plethora of age-associated diseases, including Alzheimer's disease, osteoarthritis, kidney disease, and so on. I just have a few papers here. And the question that is interesting for us, what is going on with senescence in human skin aging? In this respect, we published about 20 years ago a study where we show that in healthy human people, we see the accumulation of senescent cells as the people are aging. So here we have a young donor, and we see senescent cells. So they are P16 positive cells are here. Senescent cells are showing up in the epidermis and in the dermis. And in the old donors, you have an increased number of these senescent cells. And here's the quantification. So we see that in the very old people, like up to 95 years, there you can have up to 10% senescent cells in your skin. But in the lower age groups, it's usually between 1% and 5%. So that's a minor percentage of senescent cells. And we think these cells are driving the aging process. Skin aging is also enhanced by sunlight. You can see this on this picture of a truck driver who experienced 25 years of sun exposure on the left side of his face due to his open window during driving. So skin aging is enhanced by sunlight. And so this led us to the hypothesis that UV-induced senescence of skin fibroblasts may drive skin aging, which is the basis of the rest of the story I'm going to tell you today. In this play, we have also to consider the role of mitochondria. Mitochondria, known as the powerhouse of the cell, are prominent sources of reactive oxygen species, which are thought to contribute to driving the aging process. And it is also known that mitochondrial dysfunction plays an important role in cellular senescence in various cell types. And this led us to the next question: is there a role for mitochondria in UV-induced senescence of human skin cells? I show you the results of an experiment where we treat skin fibroblasts with mild UVB stress. In this experiment, we can recapitulate the process of photoaging, which takes more than 30 years in vivo in a 2-week experiment. And as expected, we find massive mitochondrial damage in these UV-treated human skin cells, as you can see here by electron microscopy. We see mitochondrial blabbing, we see disruption of the crystal structure. And this raised the next question: how is mitochondrial damage mitigated in these cells in order to survive? Because you will be unable to survive with damaged mitochondria. And it is known that damaged mitochondria can be eliminated by the process of autophagy. This is an important quality control process pioneered, among others, by Dr. Osumi, who received the Nobel Prize 2016 for his discoveries. And in this process, here you see damaged mitochondria. They are included into autophagosomes, which later on fuse with lysosomes, which then provide the enzymes to degrade the damaged mitochondria. And this is used for recycling to generate new healthy mitochondria. So this is a nice concept, and we wanted to see whether we can observe mitochondrial autophagy, which is also called mitophagy, in UV-treated human skin cells. And here I show you a picture where mitochondria are labeled in red and autophagosomes are labeled in green. And you see that there are yellow spots, which means these are mitochondria which are inside autophagosomes. I will now show you a small video where you see how a mitochondrion is excised from the mitochondrial network by an autophagosome and transported to the lysosomes in the periphery of the cell. So here you see the mitochondrial network in red. Here is the cell nucleus. Here are the autophagosomes. And if you look at this tiny spot, you see a red spot which has a green membrane around. And this is a damaged mitochondrion, which is now being transported from, it's taken out from this mitochondrial network, and it's going to the cellular periphery to be degraded by lysosomal enzymes. I run it once again. You see this from here to there. The lysosome we don't see in this experiment, but the case is pretty clear. So we also found that damaged mitochondria must be removed for cell survival. And in this system, we found that inhibition of autophagy changes the cell fate from senescence to apoptotic cell death. So this inspired us to think of autophagy inhibitors as new senolytics for human skin rejuvenation. And to test this idea, we are using preclinical models, which I will introduce in the next slide. So these are organoid models for the skin, which are also called skin equivalents. They are produced by combining fibroblasts in a collagen matrix with keratinocytes, which then differentiate at the so-called air-liquid interface to reproduce skin layers like dermis, epidermis, and stratum corneum. And this is a useful model for many studies in the skin, and we applied it for skin aging. A characteristic phenotype of aged skin is the thinning of the epidermis, which can be simulated in skin equivalents, as you see here, by using an increasing concentration of senescent cells in the skin equivalents from 0 to 100%. And if you look at the epidermis, you see this epidermal thinning. And we found that you can have the very same effect when you directly irradiate existing skin equivalents with UVB. So this is shown here. So this is the unirradiated cell with the healthy and thick epidermis, and then with increasing UVB irradiation, you get this epidermal thinning. In my final slide, I present you an experimental outline of how to test small molecules for their rejuvenation capacity in skin equivalents through the elimination of senescent cells. And this is, I'm leading a consortium where we want to do exactly this, and we are in the middle of our work, and we look forward to results to come very soon. Here I have the acknowledgements, and I would like to discuss points which are similar to what Pidder has already talked about. So question is, what happens after senolysis? And he may have a good answer to this. Second question in the field, is it good to remove the senescent cells altogether, or would it be better to just make them from bad neighbors to good neighbors? That would be done by xenomorphics. We can discuss this, expand this in the discussion. And potential applications for us would be threefold: cure age-associated skin disease, prevent skin disease in healthy elderly, or delay skin aging in healthy adults. And with this, I thank you for your attention, and then we can enter the discussion. Thank you.
Thank you so much. Two very interesting presentations on, of course, a fascinating topic. I think there is some consensus to the point that both of you seem to believe that the treatment with senolytics can revert at least a few aspects of skin aging. The fur that Peter de Keizer showed, the fur frailty decrease, can be considered to be a skin, aging reversal to some extent. The thing is that the skin, of course, has different compartments: melanocyte compartment, keratinocyte compartment, fibroblasts, and kind of progressing or following through on the question that Professor Jansen-Duerr already presented on his last slide. Senolytic treatment, would there be differential effects on these three different compartments, as they have different kinetics and replenishment of stem cells potentially? So would you believe that the senolytic treatment would have beneficial effects on either of these three compartments? Maybe Peter de Keizer, could you perhaps comment on this?
Yeah, the honest answer is we don't know, so we can only speculate. But I think a few important points is that what is senescence, right? So I think not all senescence is equal. I think that we also started to realize. So first of all, within the cell type, you have difference in senescence. But what you now describe is if senescence in a melanocyte is different from a senescence in a fibroblast, and if that would matter. I think you're right. We generalize too much, and I think not all senescence is equally bad. By which I mean that some senescent cells probably still have a function, and they actually removing them may be bad. There's some evidence also again in the liver and in pancreas that removing P16 high cells is actually doing more harm than benefit. Again, benefit. So the honest answer is I don't know. We would have to experimentally address this, and for that we need to have more specific senolytics. So those that only kill the senescent melanocytes, or those that only kill the senescent fibroblasts, which we don't have at this point. Like they're all kind of considered senescent, which is not true. We are working on that. Like I said, what I showed you, this compound was very specific for a subtype of senescence, female high scarred senescence, as we call it. And yeah, that is very much DNA damage high, basically, that type of senescence. So yeah, long answer to say is we really have to do this experimentally. We don't know.
Professor Jansen-Duerr?
Yeah, that's, I agree to what Pidder says, but there is another interesting phenomenon which I did probably not pronounce too much in my presentation. This experiment where we take senescent fibroblasts, put them in the skin equivalent, and we see massive effects on the epidermis. And we see also more interesting effects than is shown in this Senescence model, which I was showing, gives you the idea that there's a signal coming specifically from the senescent fibroblasts in the dermis, which instructs the epidermis to generate H-like phenotype. And this is a very interesting topic. There you may ask, what could be these molecules, or what could be the signals like? And there are, of course, SASP components, and the SASP, as we know, is comprised of many cytokines and other protein-like molecules, but you also have more complex structures like extracellular vesicles. And my feeling is that the fibroblasts at least in skin aging, the senescence of the fibroblast is, in my view, most likely a very potent, if not the major trigger, which instructs the rest of the tissue what is going on. I mean, we have also seen senescent cells in the epidermis, and we now know that these are mostly melanocytes. Of course, we don't know whether the melanocytes signal back to the dermis, but from the setting of this experiment, where you just take normal keratinocytes with senescent fibroblasts, you change the entire skin equivalent architecture. I would argue is a good argument to really try to figure out or identify the fibroblasts, the few senescent fibroblasts in the dermis as the culprits which drive the process. That's, of course, a strong hypothesis, and we have to prove it. But I have the feeling that that could be a good way to pursue.
Right. Thank you so much. I think with this, we're almost at the end of the time allotted to the discussion. A lot has been clarified already. I'm not sure, Neil, do you want to add any comments or questions?
I think a fascinating topic, and I think if we had like another half an hour, we could go into many aspects. So thank you so much for that. We have one question from an attendee, which either of you may choose to respond to, and that is, what about the side effects of senolytics? What about the approach of using extracellular vesicles, a new part of the SASP, as a target? But I don't know whether you want to respond to either of those.
I think in terms of side effects, it's surprising actually that we have observed very little side effects so far. Like all the tens of senolytics that have been published, and I've also done these experiments myself. Back in 2011, I think it was the first paper from Jan van Deursen who did the genetic ablation of senescence, right? And it was in a very fast-aging Bubber1 mouse. I thought it would die because you have a small animal that is already very stressed and damaged, and then quite radically you remove cells from the animal. It did better so far, so it seems to be okay. I think that the difficulty, of course, is in like a burst. If you create an apoptotic and pro-inflammatory burst, so this is something you probably have to mitigate. But other than that, so far, what we've seen in our mice is that it's very well tolerated. The lab that used our compound said it's like injecting PBS. Like it's not really much. They don't really look sick, at least. But that's all phenotypical, of course. And last part about the extracellular vesicles, I think that's very, very interesting also from a diagnostics point of view, because senescent cells secrete things into the bloodstream, but of course that's very much diluted out, right? Because you don't know if IL-6, if it comes from your small toe or your left earlobe or your kidney or whatever. But vesicles, you could potentially, hopefully, also isolate vesicles from senescent cells that are unique for a specific subtype of senescence or cell type. So my hope is actually that we will see much more of this in the future. I think I'm not sure what the question was about, if we can target it, because that I'm not sure how that would work. If we use it, I think we can use it for diagnostic. Let's keep it at that. Yeah.
Perfect. Pidder, do you want to add anything to this?
Yeah, I want to add something.
Please.
I would be a little bit more careful concerning the side effects of the senolytics. I mean, Pidder has for sure brought the right argument concerning the genetic elimination of the cells, because this is for sure targeting P16 positive cells. But nowadays we have a lot of compounds around which are named senolytics, and they have some effects, and you can read tons of papers on senolytics which do this and that to fight aging in many mouse models. I'm not sure in each of the cases that the specificity of these cells for these compounds for senescent cells is 100% established. So I'm a little bit more hesitant here to say, hurrah, we don't have any side effects, because in some of the assays that are used with mice aging, you can easily get lost what is a side effect and what is a real anti-aging effect. So
It's true for any compound, right? I mean, all compounds have side effects at some point, so that's not different. I think that's maybe more due to the compound than the senolysis part.
Yeah, exactly.
You're right. I mean, we have to assess that in proper clinical trials. And, you know, we just raised money to actually do this in monkeys. To actually go from we did it in mice. Now we have to go to rats, and then in January we'll have data in monkeys. Then we should know if this is a no-go, let's say.
So our strategy would rather be to go to the human skin, because then you have topical application and you have a better chance for ethical permissions. But this is, of course, still in the future.
Yeah.
Pidder, I think
Oh, sorry,
I was going to say, Pidder, that image that you shared of the I believe the truck driver with the one side that is internal control for the other side. Imagine if we could reverse that topically. That would be fantastic. I will plant that seed in your minds, and I want to bring the session to a close now and thank both our fantastic speakers for their tremendous contributions. Firstly, the generosity of their time, despite running such busy labs and doing everything else, to come and speak to us at lunchtime and share their views. So we thank you both very much. We now need to make some minor announcements about our next upcoming session, and I invite Florian to share the slides. So everything that we've said here and the discussion is recorded, and we will soon have these available on YouTube. Next slide, please. The next session that we have coming up is 3D bioimage analysis of volume EM of skin cells by Dr. Ross Laws, and that will be on the 17th of July at 1:30. Next slide, please. The upcoming ESDR meeting in Lisbon, Portugal will be fantastic. And even though abstracts submissions recently closed, we are accepting late-breaking abstracts from the 1st to the 19th of July. And please also note, to get there at the best rates, consider our early bird fees. And finally, please consider joining the ESDR. We have some great promotional aspects at the moment. Remco, I don't know whether you want to add to any aspect of our membership opportunities.
Well, I would encourage all academic dermatologists and skin researchers to become member and also to attend the Lisbon meeting. Do not have any specific additions to make. Thank you, Neil.
Perfect. Well, with that, everyone, thank you, and thanks again for your time, and have a good day. Bye for now.